A recent study found that breathing 5% carbon dioxide in short pulses may improve amyloid and tau out of the brain of people with Alzheimer's. That is an extraordinary result, but I believe the researchers may have demonstrated only half of what carbon dioxide can do. Their protocol focused on pumping fluid through the brain by opening and relaxing the cerebral blood vessels. There is another pump. It may be just as important. It may be even more powerful. And carbon dioxide activates it every time it makes you take a deep breath. The study came from researchers at the University of Mexico, and participants alternated 35 seconds of a medical gas containing 5% carbon dioxide and 35 seconds of room air. They repeated that cycle across 3 sessions lasting about 10 minutes each. When carbon dioxide rose, the brain's blood vessels opened. When participants returned to room air, those vessels moved back towards baseline. Open, return, open, return. That rhythm created a vascular pump. MRI showed that the vascular movement was tightly coupled to cerebrospinal fluid entering the brain. Blood tests then showed temporary increases in amyloid beta and other brain-derived proteins. That is consistent with waste moving out of the brain and into the bloodstream for disposal. Now, I want to be very precise about what this study showed. There is evidence of a clear mechanism. It was not proof of an Alzheimer's treatment or cure, but the mechanism is important because the brain has a waste removal system. It's called the glymphatic system. Cerebrospinal fluid moves through spaces around the brain's blood vessels. It mixes with the fluid surrounding the brain cells and carries metabolic waste toward the exit. This system becomes especially active during deep sleep. Most explanations focus on the arteries. As an artery expands and contracts, that movement helps push fluid through the surrounding space. That is the pump the study deliberately activated. But cerebrospinal fluid is not moved by the arteries alone. Breathing moves it too. The diaphragm is a large dome-shaped muscle beneath the lungs. When you inhale deeply, the diaphragm drops. Pressure inside the chest falls. That creates suction. When you exhale, the pressure reverses. That creates more of a push. So you have suction, then push. And this pressure wave moves venous blood back toward the heart. It drives lymph through the thoracic duct, and it also pulls cerebrospinal fluid upward from the spinal canal and sends it through the brain. This is not a minor effect. In an awake human MRI study, deep abdominal breathing increased the upward speed of cerebrospinal fluid by 28%. But the researchers measured something else too. Imagine water moving through a channel in a series of waves. One measurement tells you how fast the water travels. That is the 28% increase. The other measurement tells you how strongly the water surges in rhythm with each breath. That breathing-driven surge increased by 118%. It more than doubled. Both numbers describe the effect of deep breathing. One measures the speed of the fluid, the other measures the strength of the repeated wave created by the breath. During deep abdominal breathing, respiration became comparable to the heartbeat as the driver of cerebrospinal motion. Other awake human MRI studies found the same basic pattern. Deep breathing increased fluid movement throughout the brain and spinal canal. It also synchronized cerebrospinal fluid with venous flow. The breath is a pump. And here's the alternate hypothesis I believe deserves serious attention. The New Mexico team used intermittent carbon dioxide to create artificial rhythm in the blood vessels. But a continuous therapeutic level of carbon dioxide creates another kind of rhythm by stimulating the drive to breathe. As carbon dioxide rises, the brainstem signals the diaphragm to work more deeply and powerfully. Every deep breath then produces a larger swing between negative and positive pressure. That means that continuous carbon dioxide may drive glymphatic flow through the diaphragm, even without repeatedly switching the gas on and off. The intermittent protocol emphasizes the arterial pump. Continuous carbon dioxide may emphasize the diaphragmatic pump. And deep carbon dioxide breathing may engage both. That is not speculation pulled from thin air. Every link in that chain is supported independently. We know that carbon dioxide stimulates the respiratory drive. We know deeper diaphragmatic breathing produces larger thoracic pressure oscillations. We know those oscillations move venous blood and lymph. And awake human MRI shows that they dramatically increase cerebrospinal fluid motion. What has not been performed is the final experiment, connecting continuous CO2 enhanced breathing directly to amyloid and tau clearance. That missing experiment does not invalidate the mechanism. It identifies potentially the next study that needs to be done. Some researchers have raised a concern about continuous carbon dioxide. They cite animal experiments in which sustained hypercapnia reduced glymphatic transport. But look closely at the experiment and you'll see that those mice were anesthetized. In the controlled hypercapnia condition, they were intubated, mechanically ventilated, and chemically paralyzed. They could not respond to carbon dioxide by breathing more deeply. Their diaphragmatic pump had effectively been removed from the experiment. They were also given 5% carbon dioxide mixed with 95% oxygen. That is very different from an awake person breathing a therapeutic CO2 mixture and responding with natural respiratory movement. The mouse study tells us something useful about sustained vascular dilation. It does not answer the question I am asking though. What happens when an awake human breathes carbon dioxide deeply enough to turn the diaphragm into a powerful fluid pump? So my explanation is clear. The stronger pressure oscillations should increase venous return, lymphatic propulsion, and cerebrospinal movement. That increased movement should support the clearance of amyloid, tau, and other metabolic waste. The exact dose and protocols still need to be tested, but the physiological argument is strong. There's a deeper reason this matters though. Amyloid and tau are not simply innocent debris. Once they accumulate, they can damage tissue, amplify inflammation, and make clearance even harder. Removing them is a real therapeutic target, but proteins do not begin misfolding for no reason. Cells require energy to fold proteins, repair them, and dispose of damaged ones. They require stable chemistry, good circulation, restorative sleep, and functioning mitochondria. When that terrain fails, more proteins misfold and fewer proteins are removed. Then the waste further damages the terrain. It becomes a vicious cycle. So carbon dioxide may intervene on both sides of that cycle. It can support waste movement through the vascular and diaphragmatic pumps. It can open cerebral vessels and improve microcirculation. And through the Bohr effect, it helps hemoglobin release oxygen into the tissue that needs it most. So CO2 is not the fuel. It is the coordinator of flow. This does not mean that anyone should improvise with concentrated gas bags, tanks, masks, or enclosed spaces. 5% carbon dioxide is a powerful physiological stimulus. concentration, oxygen balance, individual health, experiment, and monitoring all matters. The responsible conclusion is not to copy an experimental protocol, it's to test a complete mechanism. Compare intermittent carbon dioxide with continuous carbon dioxide. Measure breathing depth with diaphragmatic movement. Measure the vascular pulse and the respiratory pulse separately. Measure which protocol removes the most cerebrospinal fluid and clears the most brain-derived waste. The recent study showed that carbon dioxide can switch on a brain clearance mechanism while a person is awake. That alone is a major discovery, but it should not narrow our thinking to one timing pattern or one pump. The arteries move fluid, the diaphragm moves fluid. Carbon dioxide influences both. The study proved the first pathway can work. The existing physiology gives us a strong reason to believe the secondary pathway can work too. And the most powerful protocol may ultimately be the one that brings both pumps into rhythm.